Deep inside your brain, there is a biological machine built to last a lifetime. Unlike your skin or blood cells, which constantly regenerate, your neurons are with you for the long haul. They are post-mitotic, meaning they cannot divide to dilute damage or shake off infections. To survive for decades, they depend entirely on a sophisticated internal logistics and quality control network. But what happens when this network fails? Today, we are exploring how the breakdown of this system is the root of neurodegeneration and how a class of sophisticated invaders, neurotropic viruses, have learned to hack the machine. To understand the crime, we first have to understand the system under threat, neuronal autophagy. Think of this as the neuron's digestive tract and recycling center combined. Because neurons have incredibly long axons, this is a massive logistical challenge. The process often starts way out in the periphery of the neuron, but the actual degradation happens in the cell body. This reliance on long-distance transport creates a supply chain with multiple points of failure. And, as we are about to see, viruses have evolved to exploit every single inch of this highway. Meet the saboteurs. These are neurotropic viruses, and they don't just crudely smash the machine, they hack it. They have evolved sophisticated molecular strategies to manipulate the autophagic machinery. It is rarely simple inhibition. Instead, they dissect the pathway, hijacking the early steps to build their own replication shelters while dismantling the final stages to avoid being eaten. Each family has a unique method of operation, targeting specific choke points. Let's examine our first suspect. First up is herpes simplex virus 1. We call this one the insider because it uses a strategy of sequestration and deception. Its primary weapon is a neurovirulence protein called ICP34.5. This protein acts like a master key that binds directly to Beclin-1, a critical initiator of the autophagy process. The outcome is an absolute blockade. The virus effectively handcuffs the security guard before the alarm can even ring. We know this is intentional because viruses modified to lack this specific binding domain show dramatically reduced virulence in the brain. But HSV-1 isn't done yet. It also launches a secondary attack on the neuron's power plants, the mitochondria. Normally, when mitochondria get damaged, the cell clears them out in a process called mitophagy. HSV-1 cripples this process for a sinister purpose. By preventing the degradation of leaky mitochondria, the virus stops mitochondrial DNA from spilling into the cytosol. That floating DNA would be a massive danger signal to the immune system, so the virus essentially hides the trash to avoid alerting the police, leaving the neuron rotting with accumulating metabolic damage. Next up, we have the Zika virus, whom we call the factory builder. Zika plays a dangerous double game. First, it actually induces the early stages of autophagy using proteins like NS4A and NS4B to hijack the machinery. It forces the cell to build vesicles, not for cleaning, but to use as replication factories for its own RNA. Then, it executes step 2. Arrest. It blocks the fusion with lysosomes. It effectively transforms the autophagy pathway from a waste disposal system into a viral production line. If Zika is a corrupt builder and enteroviruses are demolition experts, their strategy is proteolytic sabotage. They carry viral proteases, essentially molecular scissors specifically designed to snipe the cellular machinery. Look at the mechanism here. The virus targets SNARE proteins, which are the zippers that allow waste vesicles to fuse with lysosomes. The virus simply snips the zipper. Meanwhile, it tricks the cell into ramping up autophagy upstream, creating a futile cycle. the neuron is trapped, generating waste it can no longer clear. Let's pause and look at the big picture. While the methods differ, the target is the same. We have HSV1, the insider, freezing initiation to hide. We have Zika, the factory builder, hijacking the middle steps for construction. We have Enterovirus, the demolition expert, cutting the cords at the end. And we even see HIV1 acting as a bystander, locking down the pathway via Nef and TAT proteins. Every major neurotropic virus has a file on how to break this system. This leads us to the Convergent Catastrophe. Regardless of the specific viral strategy, the result for the neuron is identical. The cell becomes choked with its own undegraded waste. We see mitochondrial fragmentation and toxic sludge building up inside the cell body. This is the crucial link. This mechanism bridges the gap between an acute viral infection, like a temporary fever, and chronic long-term neurodegeneration. The virus might leave or go dormant, but the damage to the waste disposal system remains. The evidence is in the fingerprints left at the scene of the crime. We are now finding specific viral signatures in major neurodegenerative conditions. In Alzheimer's, we know that HSV1's inhibition of Beclin-1 directly impairs the clearance of amyloid beta plaques. In ALS, the enterovirus protease cleaves nuclear pore proteins, causing TDP33 to leak into the cytosol, exactly mimicking the core pathology of ALS. These aren't just coincidences, they are mechanistic causes. But understanding the sabotage gives us a new playbook for fighting back. Instead of just trying to kill the virus, we can try to fix the machine. If the problem is enterovirus cutting proteins, we can use protease inhibitors to stop the scissors. If the problem is stalled flux from Zika, we can use mTOR-independent inducers to jumpstart lysosomal biogenesis. We are moving from simply antiviral to host-restorative therapies. However, there is a critical caveat here. Timing is everything. Remember how Zika uses early autophagy to build its factories? If we blindly induce autophagy during early infection, we might actually help the virus replicate. But if we intervene in the late stage, inducing autophagy while simultaneously restoring lysosomal function, we can clear the pathogen. The goal isn't just to turn the machine on, it is to restore autophagic flux, the complete flow from creation to destruction. This brings us to our unifying principle. The subversion of neuronal autophagy is the mechanism that explains how diverse viruses cause both acute encephalitis and potentially trigger chronic neurodegeneration. They turn a vital defense system into a vulnerability. By mapping the lock, the factory, and the broken gear, we uncover a new path forward. Restoring the flow of neuronal autophagy represents a paramount strategy for treating not just the infection, but the devastating neurological consequences that follow.